Hey folks, welcome to the IOB faculty seminar. I'm Zarif, and today I have the pleasure to introduce SM Ashiqul Islam. Dr. SM Ashiqul Islam is an assistant professor at the University of Albany, SUNY. He has a joint appointment at the Department of Epidemiology and Biostatistics and the AI Plus Institute. Ashiqul received his PhD in biomedical studies with a focus on bioinformatics from Baylor University. His experience is unique since he had also the experience in the industry before joining academia. He worked as a staff scientist at Thermo Fisher Scientific. Ashiqul's research focuses on delving deep into cancer genomes to translate the knowledge for healthcare applications. To this end, he develops AI and machine learning tools to analyze multi-omics data. He has published several software in this area. So I have known Ashiqul for a while, since we went to the same school. We went to school in the same place for our bachelor's and master's, and he guided me throughout my PhD application process and had an important role in making me passionate about bioinformatics. So without further ado, please join me in welcoming Ashiqul.
Yeah. So thank you very much, Jarif. Um, can I start my talk?
Yeah, you can start your talk, but you have to share your presentation first.
Yeah, sure. So can you see my screen?
I can see your screen, but there are also these transcripts on the bottom of the screen. So
Okay.
yeah,
Maybe we need to, I don't know how to close it.
I mean, you can try, and if you're not able to do that, I think we're good to go with this format.
Okay. Yeah, I'm not quite sure how to.
I mean, if that's
Okay.
the case, the transcripts are okay. I mean, that's the best we can do right now. If you are in person, that would be a different thing, but you're good to go.
Okay. So thank you very much for inviting me to give a talk in the bioinformatics department at so is it the bioinformatics department or
It's the Institute of Bioinformatics
Institute of
at UGA.
okay, okay. So Institute of Bioinformatics. Thank you very much. So today I am going to talk about the research I did during my postdoc because after doing my postdoc for the next 3 years, I could not do any research, and then I just join at the SUNY Albany, and I'm still starting new research here. So today I will mainly focus on the research that I did in UCSD, like University of California at San Diego, and that is about revealing the hidden language of cancer genetics through a holistic approach to deciphering genomic alteration. Okay. So today's content will be grouped into 3 chapters. In my first chapter, so in the first chapter, I will little bit talk about my research background. Then I will directly jump into the chapter 2, which is the main content of the presentation of today's presentation. And this is about the development tools to uncover mutational signature in human cancer genome, and that will include definition. So, what is the mutational signature and the motivation behind the work? Then, how AI is involved in this work, and then the the methodology development part, and then the result. Then in chapter 3, I will briefly talk about the future research agenda or my future research plans. And yeah, so here is the first chapter. So my research background. as Jarif told that I did my PhD. I completed my PhD at Baylor University, where I worked on machine learning based like methodologies and tools to predict protein characteristics from their primary sequences. So that was my first exposure to the application of machine learning to in bioinformatics area. Before that, I was working in the wet lab part, but since then I got involved in the artificial intelligence or machine learning based area. And there I worked with two different advisors. So instead of having one advisor in the PhD, in my PhD, I had two advisors or two supervisors. One was from the Department of Computer Science, another was from the Department of Biology. Not biology, it's from like molecular biology. Then after completing my PhD in 2018, I joined at UCSD, so University of California, San Diego, as a postdoc in 2018, and there I fully involved with the application of machine learning on human cancer genomics. And there I published a couple of papers, made a tool, and that's what I'm going to actually present today. Then in 2021, to get some experience in the so to get some industry experience, I joined at Thermo Fisher Scientific in California, Silicon Valley. And there I developed machine learning based methodologies and tool to use methylation array. Uh, I think I have changed my slide little bit. So can I stop this one and share the new one? Because I made some modification and I cannot see that here. Okay. So please let me um, stop this and share the new one again. Um, can you see my screen?
No, I don't see any screen. Probably you unshare it and share maybe your whole desktop and then go to the presentation. That way, I think
Yeah,
we won't also see the transcripts.
sure. Because I did some modification and I could not see that modification in my PowerPoint. Now can you see?
Yeah, I can see the presentation all right. Like
Okay, okay.
chapter two and stuff. Okay. Yeah.
Yes. In the industry, um, I worked on development of methodologies and tools to analyze methylation array data, and also I was working on pipeline development on image processing. So there, my role was somewhat different than it was in the academia. In company, the research was mainly, so I was in the R&D team, but they were doing the research on trying different camera system, trying different chemical system, and my role was to develop pipeline to analyze their data. So my role was to work as a data analyst there, and like the pipeline developer or developing new pipelines to analyze data. The difference in industry was I could not introduce my own methodologies or whatever. I could not explore new thing. I just had to introduce the things which are very which were already published or routinely used in other places, and I just had to make the pipelines there to analyze data. So nothing, no new investigation or no new exploration there. Okay, in the industry. Now, in postdoc, so I'm going to talk about my postdoc project, and that was on uncovering novel mutational signature in cancer genome. So if you don't know what are mutational signatures, those are the elemental patterns of genomic alteration that can be used as biomarkers. Okay, so um, unlike with the mutational signatures, we can characterize a genome in a holistic manner. So unlike a point mutation or SNP, what we associate with some clinical trait, here in mutational signature, we try to capture the whole pattern of the mutations in a cancer genome and use the pattern of the mutations as biomarker to relate their clinical traits, disease, and other exposures of a cancer genome or of a cancer patient. The the use of this kind of this procedure is, if you, so there are many noise in the genome. So if you don't capture the point mutations or the point alterations, the genomic pattern, so these biomarkers are very robust, and that can sustain to noise. And we can, so the bottom line is we can actually capture the genome in a holistic manner. Now, here is the motivation that if you consider the development of a tumor-resistant cancer genome from a fertilized egg, so in different steps, the genome can accumulate mutations, and those mutations can come from different exposure or different causes or different processes. Then in the final stage, we can have a big number of mutations in the cancer genome, and our motivation is, can we separate the mutations those are coming from different processes? Like here, some mutations are coming from process A, those that can be, for example, genetic defect, like germline genetic problems, or from smoking, or from some chemotherapy. So these are the process which causes mutations in the cell throughout its life, and then it finally creates um, a group of mutations in the genome. So the motivation is, can we separate the group of mutations those are coming from different processes? So that is the question. And if we can answer the question, then we will be able to better understand the mechanisms of cancer development. And then we also can use those for personalized cancer treatment, and also epidemiological studies. If we can answer those things, like we can separate the group of mutations and relate them to the um, causal things. Okay. And now the question is, can we do that? Can we do that? So can we separate the signals from the mixture of the signals? And the question is, yes, we can do that mathematically using blind source separation. And um, the concept of a concrete concept of blind source separation is um, here. So I tried to demonstrate a concrete example of the blind source separation. For example, you have a mixture of sound, those are coming from different instruments, okay? And then using blind source separation, we can estimate or separate the signals, those are coming from different instruments. And that is the blind source separation. And how can we do that? To do that, we need we need multiple examples of the sound mixtures, and then we can use AI methodology or AI tools to do the blind source separation. You can use PCA, you can use independent component analysis, SVD, or non-negative matrix factorization to do that. So anything. And we can do the estimation. So we can separate the signals from the mixture, or the components from the mixtures, and then we can relate these signals to its source instruments based on statistical analysis. Okay, so that is the concept of the blind source separation. And in our case, instead of the mixture of signals, we have the group of genomes which has the catalog of different mutations. So here we have six different mutations. To make the example easy, I kept this one very straightforward, like only six features. So if we know in the genomes, and if we know the counts of each type of mutations from in different cancer genomes, then we can do the same strategy. So we can apply the same strategy to separate the patterns or the components, what we call signatures here, the mutational signatures. So that actually tells in one mutations what are the probability of coming that mutations from the six different classes of mutations. So that is so in reality we have more classes. I'm going to explain that in a minute, but that is very simple example. That is like mutational signature 1, that is 2, this is so we can extract it up to n. And then if we have the clinical traits or clinical data of each of the genome, then we can statistically associate these signatures and use them as a biomarker to different process like smoking, chemotherapy, or UV ray exposure. That could be different process or different causes. So that is the concept of the blind source separation of mutational signatures. Now, as I was talking about the six mutational classes or the features we use. So if you are so if you know what is machine learning, then if you are familiar with machine learning, then you know the features, like what are the features which we use to classify a so a like something, okay, a data. So these are the features, and we use the six mutational classes as features basically, but not exactly the six classes like C2T, C2A, C2G, and T2A, T2C, and T2G. But we we actually use the 5' and 3' nucleotide. Then we make a mutation type. So not based on that six mutation classes, but we capture the base of the 5' nucleotide where the mutation is happening, and the 3' nucleotide. And in this way, for each mutation class we get 16 mutation type, and for the 6 mutation class we get 16 into 96 mutation types. So these are our features, and using these features, our sample or the genome catalog look like this. So this is a matrix. So we have 96 types of mutations, and these are the specific genome or patient. So we actually compute from a VCF file, we compute the number of mutations. These are coming from each mutation types. And this is a classic example of a mutational signature. So the x-axis is the 96 different mutation types, and the y-axis, or the percentage of happening of happening a mutation from each of the mutation, like 96 mutation types. So it is SBS4 that we also called the smoking signature. So that is a signature, okay? And we have associated this signature to the in lung cancer, to like tobacco smoking. And here we can see the a predominance of C2A mutation, because smoking causes a G2A mutation, because there are polycyclic aromatic hydrocarbon in tobacco, and like benzopyrene, and that can actually interact with the guanine, or G, and then it actually mispair the guanine with so instead of C, it mispair the guanine with a T, so thymine. That's why we have a C2A mutation, and hence we can see a predominance of C2A mutation in the smoking signature. Okay, and this signature we can actually use as a biomarker to associate it with the tobacco smoking. So if you analyze the mutational signature of a person, and if you can see this signature in that genome, then you can easily tell that, okay, this person actually smokes. So that is the concept. Now, like the tobacco smoking, we have extracted a few more signatures from different tissues, and we have these in the cosmic database, and those signatures are associated, like this is apobic signature SBS2, and there are so we were able to connect these signatures, each of the signatures, into different etiologies, like different drug exposure or different chemical exposure. Then um, as I told, that the mutational signatures are equivalent to the principal component we get from PCA analysis. But here, to extract the signatures, we can use PCA, but we don't use that, because PCA can give the negative values, and it is very difficult to explain the negative values in terms of to explain the mutation events in our genome. So for the sake of the explanation, we use non-negative matrix factorization, which is another algorithm which work with non-negative data and also generate the non-negative components. Okay, so my own project goal there, that was my biggest project. I was involved in many other projects, but that was my biggest project, and I was the first author of the publication of this project. So that was about developing a methodology and tool that can accurately extract mutational signatures. And to do that, I have developed a tool that is called Seq Profiler Extractor. So that is a tool, and it is a Python-based tool. You can also get that in R, and so we have library in GitHub, and also there is a library you can download that with pip install Seq Profiler Extractor. So that is the workflow. That is the main workflow of this tool. So first, we have the VCF files or the map files from the genome sequencing as input, and we can make the metrics, and the metrics look like this. So that is the first step, and the metrics look like this. So as I already showed you, the rows are different mutation types, 96, and the columns are different patients. And then we do the matrix factorization step. Then we select the correct number of signatures, because we have to interrogate with different number of latent component, but at the end of the day, we need to see the optimum like number of signature, and then step 4, we get the signatures. Okay, so in step 2, as I told, that we use non-negative matrix factorization to extract the signatures from the cohort of cancer or the mutational catalog, and we need to try the component equal to 1, 2, 3, 4 up to Z. So it is normally 20. If we see if we can see that there may be like more than 20 signatures, then we can extend the interrogation. But normally we interrogate up to 20 signatures. So we do that for each number of signatures. And here we have an example of the non-negative matrix factorization. So that is the cohort or the catalog. Then after applying the non-negative matrix factorization, we have the signatures. Here we have, in this particular example, we have interrogated with for 4 signatures. So that the W matrix are the signatures, and the H matrix, after doing the matrix factorization, the matrix H is the weights of the signature, where um, these are the 4 signatures rows, and the columns are the weights of each signatures in each genome. So we can see that, okay, what signature is present in a higher intensity in a particular genome. Okay, so that is the weight matrix. And we do that 100 times after putting Poisson noise. And I'm going to tell that why we do that 100 times. And that is basically to increase the robustness of the tool or of the system. So that is the sample data set. And after doing that interrogation, that is the step 3, the model selection. So here in this step, we have a number of algorithms that operate to select the number of signature based on the stability, and which is a partition-based clustering and silhouette coefficient, and also based on the reconstruction error. So what the stability actually tell us, so if we extract that, so after putting the noise, Poisson noise 100 times, so creating this type of 100 X matrix or the catalog matrix, and if we extract the signature 100 times, how many times we are getting the similar signature? That is the stability. So that is the concept of stability. So if we get different signature in different times, that means it is not stable, it is overfitting. So we are getting different signature in different times. So if we get the similar signature most of the time, that means the stability is high. And then the reconstruction error, that is very simple concept. So that actually increase the sensitivity, but if the reconstruction error is too low with a low stability, that means it is overfitting, and the signatures or the components are over splitting. That means those are not stable. So we want to keep in a situation in a number of signature where we get a good stability, that means good reproduction of the signature in different trials, and a low reconstruction error. And based on these 2 matrices, we select the number of signature. And in this particular example, we have selected 3 signatures. Okay, so that is a theoretical or hypothetical example. Uh, Now, after doing that, we have step 4, which is printing the signatures, and we do many other things which is out of scope to explain in this talk. But we do the association. That means we actually clinically associate these signatures to different etiologies. Okay, and that is the basis of the tool. That what the tool does, and that is the result of my tool. So how does we have some other tools published to do the similar type of work, but my 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 PhD, my postdoc supervisor, whose name is Ludmil Alexandrov, he was the first person who extracted, so who came up with the idea of mutational signatures, and he published a tool. Then I just improved the tool, okay, with new algorithms. So in addition to the old tool, under his supervision, I also compared the tool I created or developed with some other tools, and then I had to compare. And to compare the tools, what we did, we have extracted signatures. So first we simulated some mutation, so some mutational catalog or genome cohorts with known signatures, and then without and with noise, and then we extracted signatures from those simulated cohort and compared the sensitivity, precision, a false detection rate, and F1 score with my tool as well as some other tools. And that is the result part, what we got. So that was my tool, Seq Profiler Extractor. That was the tool that my PI developed in the first place. And there are some other tools that is developed to extract mutational signature. So first I compared the tools with the noiseless data, and there's the based on the sensitivity. So how good the tools based on the sensitivity, and the sensitivity is so that is the definition of the sensitivity. It is true positive divided by true positive plus false negative. There is the definition of the true positive, false positive, true negative, and false negative. So here the sensitivity, we can see that Seq Profiler Extractor had a sensitivity around 85%, while the other tools were not as good in terms of the sensitivity of the known to extract the signatures from the simulated data. And then the false detection rate, here is the definition. And we also found that Seq Profiler Extractor was, so that the false detection rate was around 2.5% with Seq Profiler Extractor, while the other tools detected a lot of signatures which are not correct signatures. Okay, so that is another matrix, and that is the F1 score, which is a harmonic mean of the precision and sensitivity. And for this matrix, we also can see that Seq Profiler Extractor was very good for F1 score compared to other tool. Now, like other tools, we also wanted to see how robust the tools are if we put noise. So we put different level of noise, like Gaussian noise, in the cohorts. So in the simulated mutational cohorts, so 0%, 1%, 2.5%, 5%, and 10%. And we were able to see that, okay, in terms of precision, so Seq Profiler Extractor was very good at precision at each level of the noise, but the other tools were kind of like breaking down at 5% or 2.5% of noise. Same thing happened in sensitivity. Seq Profiler Extractor performed much better than the other tools in terms of sensitivity. So the results and outcomes, sorry. So the results was like we were able to develop a tool which has a better sensitivity, precision, and F1 score compared to other tools in both noiseless and with noise data. We were also able to extract 4 novel signatures from the ICGC or the PICWAG data set that is comprised of 2,700 data, and Then we were able to um, also we were able to link one of the signatures with smoking signatures. Um, uh, uh, yeah. And then this tool is also available. So as I told that Python, R, and Bash, and that is also GPU enabled. So it is pretty fast tool compared to other tools. And that was published in Cell Genomics, and I was the first author of that one. And it is being widely used in the cancer genomic, so by the cancer genomic researchers. So far, I got, sorry, 265 citations. So it was published in 2022 November. And since then, I have received, from this paper alone, I have received 265 citations. And it was also recognized as the one of the best. So among the 50 publications that was published in 2022 by the EGRP and the NCI. So they announced that as the like one of the most important publications in that year. And that also had some media coverage. My tool is available in the it is actually a recommended tool to extract signature by Cosmic database. Um, so that was my work, and I also have some future research plans with that. So I would like to extend the functionality of the tool with using some new feature. So the idea is, if you look at this genome, so in this signature, that actually, so this is the smoking. So that signature I actually showed you in earlier. So this signature can tell if this patient smoke or not, but it doesn't tell that what kind of medicine can be useful to this patient or can cure this patient. So it can actually track back to the causal things, but not the outcome. So I would like to use some feature instead of these 96 types of mutation types. I would like to use features that come from the positional characteristics of the genome, like where it is where the mutation is happening. Is it happening in a protein which is a dehydrogenase or a isomerase or a kinase? So I would like to capture those information and would like to extract or make signatures based on those characteristics. And then I think after doing that, we could be able to link the signatures to the future outcomes, like the drug sensitivity, which drug will work better for that patient, something like that. So that is one of my plan. I'm writing a R21 grant for this one. And also I would like to create or develop an algorithm which will more accurately, we have some algorithm on that, but I would like to create a accurate algorithm which can assign a known signature to a single genome catalog. So for example, to do the non-negative matrix factorization, we need a number of genome catalog. So only having one mixture of the mutation will not be able to separate the signals. Okay, so we need a number of examples or a number of samples to do that. But what if we have only one sample and we want to see if any of these signature what we have already extracted are in the database, if we can assign them or link them in this unknown so unknowns like sample or in the single sample. And I would like to use recurrent neural so RNN with the help of NNLS, that is non-negative least square, to do that. That is another idea. And then in other plans, I would like to I would like to continue to associate the newly discovered signatures to the clinical traits. Um, um, that can help us to associate signature with disease prognosis. So these are the plans. And yeah, that was my short talk that I had to cover within 40 minutes. But if you have some question, then please ask me about this tool or this methodology. Thank you very much. That is
Thanks so much for the
20
wonderful
minutes. Yeah.
talk. So the thing is, we'll be continuing the Q&A session 12 until 12:25 the latest. So I don't know how many people are in the Davison conference room. So Gapi, probably you're there. So if someone has any questions, uh, they can ask away, and I'll manage the Zoom questions. So for people who will be asking questions on Zoom, please raise your hand, and when your turn comes up, please unmute yourself and ask your questions. I see Dr. Summers raising their hand, but before that, Dr. Cannon asked this question, and the question was, how often do you find mutational signatures that cannot be associated with a specific cancer etiology?
That's a very good question. We find that kind of thing very often, that we cannot associate with the etiology. So it might happen from two different causes. The cause one, we don't have good clinical traits for that sample or for that group of cohort. That is one cause. And another cause is the signature is not correct. So that is not robust, so that it is kind of like it came from over splitting. That's why we could not associate that signature with clinical trait. But yeah, it happens a lot. So for example, if we have 5 signatures from a cohort, more or less we can associate 3 signatures or 2 signatures to a clinical trait, and the rest of the signatures we cannot. But also a number of times we found that we could not associate that signature in that cohort, but we were able to extract the signature again from a new cohort, and were able to link that signature to a clinical trait in the new cohort. That means the first cohort that did not have um, uh, enough clinical data to associate that signature. So the question, the in one sentence, yeah, yes, we have signature that is not associated.
Thanks so much. I'll be asking the next question from Zoom, and then we can check whether we have some from the Davison conference room. So Dr. Summers, you raised your hand. Would you unmute yourself and ask your question?
Yes, thanks. This was really great. I have two questions that are really quick fit together. One of them is, do do is your substrate that you look at strictly the proteins, or does this include RNAs? Because RNAs have got a lot of functions. And the other part of the question is, do any of these patterns include mobile genetic elements such as viruses?
That's a great question. So we don't currently we don't have. So in our current methodology, we don't have those information um, about which part of the protein it is coming from, or if it is or if it is from the RNA. So what we have actually, we catalog the mutations based on the characteristics. So count how many ACA. So here, for example, how many ACA are converted to AAA. So for in this sample, we got 58, but we have no idea that which part which part of the genome the mutation is coming from. So it only capture the number of the number of mutations of the C2A type of changes where the 5' A is, so the 5' base is A and the 3' base is A. That is the so that is the only information we have at the end.
So it
But
might be in a non-coding RNA.
yes, that might be any non-coding RNA or anywhere in in the genome. So
Yep.
we don't know where in the genome the mutation is coming from. But the proposal I am submitting that wants to capture the question that you had actually, from where the mutation is coming. And we want to make new catalogs, so new mutation types based on the origin of the mutation, where the mutation is coming from. Is it coming from a specific protein, a specific type of domain, and a specific type of RNA? We want to know the role of the part from where the mutation is coming. And when I was doing that,
95% of the genome is non-coding.
non-coding, yeah. Like 94, so 95% of the genome is non-coding. So we will get a lot of mutation from the non-coding part, but my plan to focus on the coding and the other functional part, what we can actually annotate functionally. And right now we have AlphaFold. So even if we don't know the role of the part of the DNA or part of the like protein where the mutation is occurring, we can predict that with AlphaFold. So we have this kind of uh, this kind of information right now, what we didn't have during that time we created the catalog. But yeah, my idea is to incorporate the positional or functional information in the to make the catalog.
Thank you.
And the other question was about the viral part. When we call them mutations, we actually match that with the germline DNA or the other non-cancerous tissue of the same patient. And then we call the alterations which are occurring only in the cancer tissue as the somatic mutations. So if we don't have the mutations which occurs in the germline DNA, then we don't capture. But again, we don't know if the mutations are coming from the viral DNA or not.
Or a transposon.
Yeah, or the transposon. Yeah, we don't know yet here.
Thank you.
Like we don't have that information.
Thank you.
And does anyone have any question from the conference room?
Hey, Sarif, we don't have any questions over here.
Okay, so I'm gonna ask a last question. I was just wondering, and it's more of your take on your career trajectory change. I know that you have explained that you didn't have as much independence as academia in the industry. So I was wondering, what are your other experiences and what differences or changes you are observing as a newly appointed junior professor at your university? What's the differences between the academia and industry that you are you have experienced so far?
That's a great question. So so in industry, as I told that I did not have enough flexibility to come up with a new idea uh, to um, to actually to modify the analysis what I was doing in the industry. So what they wanted me to do was to make pipelines. So we look at the papers or publication based on others' findings, and then I had to implement or reproduce the thing in our industry or in our lab. Not only me, so there was a big group. The group was coming from chemical like scientist, bioinformatician, then data scientist, then the biology people, physics people, engineering people. So all the people work together to reproduce something which was done in like well like which was published in a paper, and then we had to make the pipeline like so the reproducible pipeline in industry, but they wouldn't allow me to uh, incorporate something novel or do something do some innovation in that one because but I did not like the part because in our area things are changing very quickly. So if you want to make a good progress in that area, then you need to adopt or the new technologies. You need to use the new algorithms, AI algorithms or machine learning algorithms to analyze the data in a new way. But that was not possible in at least in our Thermo Fisher lab. So that was mostly about developing the pipeline. But here in academia, it is completely different. I cannot do anything what what we can see or what we can see it is done. We have to come with a completely new idea and merge different novel technologies to discover something new. For example, I have to here, I don't know if we change the feature types, and then we can see a good association of the new signature with the clinical trait. We don't know anything about that, but we can try it. So that is the work we have to do in academia. So we don't know if it will work, but we can try. So we need to like try that. That's the difference I can actually see between the academia and industry. So industry, you need to make sure that will work, but in academia, you can play with it. Um, but you need to get the funding, grants, all those things. So if you don't want to work with something new, or if you don't want to take challenges, then you may feel very well with the work in the industry, like with the work you need to do in the industry, because there is a similar work you need to do every time, and you will feel very comfortable with a lot of money. So that was my takeaway between the industry and academia work.
I
So hello,
think we're
Dr.
gonna.
Yeah,
Is anyone else gonna ask a question?
Dr. Summer has
Yeah, I think Dr. Summers has a
Yeah,
comment or a question.
yeah.
yeah. I put it in the chat, but it's just a quick question. Um, we're interested in the correct host or the ideal host for mobile genetic elements, particularly the ones spreading antibiotic resistance in a prokaryote system. So
Mhm,
in principle, it's a much smaller genome and very little non-coding RNA, but
mhm,
in principle, this could be applied to any organism of
mhm.
any size. Is that correct?
Yes, yes.
We'll talk later.
Actually, I'm going to work with collaborate with the Department of Health here with the similar type of problem. So what they propose to me, they want to use this kind of biomarker to find out the antibiotic resistant
Oh, wow.
bacteria. Okay.
Wow.
Yeah.
Exactly.
Yeah.
Great. Thank you.
Yeah.
So that's all we have time for. I mean, we wish that Ashiqul was in person with us and would have the opportunities to talk with the faculties as well as the students. Thank you again. Please join me in giving another round of applause to Dr. Islam. And with that, we'd like to conclude today's seminar and see you next week.
Thank you, guys.
Thank you very much. Bye-bye.